All right. Uh, so I was just asked a question before, and it's having not used this particular text here ever in a few years. This particular chapter, I mean, there's certainly a lot of similar information. I just think the Nestor chapter presents it better. I mean, I have no reason for you folks to learn about antisense RNAs, microRNAs, or ribose switches. Okay, I mean, I'm just it's interesting. It probably will be to our benefit to talk about something like the lac operon. If you've never heard about that before, that's this guy over here. Not to memorize it, but just to mention it in passing. It's kind of when we teach the micro to uh, the majors, or and certainly for people if they're going on to medical school and things like that, that's something that they have to be familiar with. It's kind of one of the ways that negative and positive feedback loops work, and sort of the way our body unlocks DNA. And so and again, you study these things in bacteria to understand what's going on in us. But for us to have to go chapter and verse on the lac operon and things like that, you'll see it in action because it happens in E. coli. And it's why in the laboratory, one of the things that you makes you aware of what E. coli does. E. coli is a tremendous lactose fermenter. And basically, what this operon does is, in the absence of glucose, which will be its primary energy source, it gobbles up lactose. Okay, and it ferments it very aggressively, and that's when you see color changes that are going to be so distinctive that you're going to look at in the laboratory. So it's sort of the secret science behind it, how it's sort of a system that has the ability to switch on and off based on availability. But so it's not something that's not understood. I just don't see the need for us here to be able to do it. So I'm going back with that rather wordy preface to this, because this particular chapter does not have a ton in it, but it's got some important stuff, particularly about the way genetic material shifts in bacteria, a little bit of the basics about mutation that I started to touch on last time we met, and then we're pretty much done with the chapter and the bulk of the rest of the time. And I'm going to try. Let's see. We have like what three? I'm looking. 3 weeks from today. It's the calendar thing. Let's see. So we get one, two. So we have basically, yeah, you'll probably get bombarded by exams toward the very end of the week of the 27th through the 3rd. Figure on one there. Maybe either the 1st or the 3rd. At least the Ides of March will not be upon you. Just restrain your joy. So, because we should be able to get this done today, probably no later than Wednesday, and be able to zip through viruses in the span of about a week or so. That kind of a thing. All right. So moving on. So much for the housekeeping part. Genotype and phenotype, very, very expressive. Really, we look at phenotype to identify things, because genotype is other than being very, very sophisticated, and that's what's on the other PowerPoint. They tell you a little bit about sophisticated techniques and how DNA technology and analysis sort of came to be. There's a really good show that I was watching, because I have an affection for British crime dramas, which was really based on a true story about the doctor, the scientist in Leicester in England, who really was the first one in the mid 80s like around 1987, to use DNA fingerprinting to identify a murder. Okay, so here are some of the notes. Terms I don't use, you should be familiar with. Okay, auxotrophs and prototrophs. To me, I don't think you need the definitions, but for instance, somebody taking a licensure exam like an NCAT, they may use the terminology, we utilize a salmonella auxotroph for what test. And the very, very famous test that they don't have in the other chapters, called the Ames test. It's a proper name, and it involves using a Salmonella oxytroph. It's Salmonella that requires a particular amino acid to grow called histidine. It's a mutant strain. Salmonella grows pretty easily. The, the the Ames test is done on any product that we either consume or utilize. If it makes it through the FDA, it has to pass the Ames test. And the Ames test is designed to see whether or not it's going to be a carcinogen. So why would you have a medication or a topical agent or something like that that potentially is carcinogenic? Well, you wouldn't. So that leads us to the Ames test. What it does is it causes a what we call a reversion mutation. It takes something that's mutated, salmonella, that has to have histidine to survive, and reverts it or back mutates it to salmonella that doesn't. What's called a salmonella prototroph. Okay. So if it's strong enough to do that, to mutate backwards, it most likely is going to cause significant mutations in us. That's the Ames test 101. So that's a really important test that, particularly for many of you who are going to be utilizing materials that are under the auspices of the FDA. Read that as medications, drugs, whatever you want to talk about. Stuff like that. Okay. So I won't use the term, but and I basically, the way I phrase the question is, what bacteria do we utilize in the Ames test? How do we, what kind of mutation? It's a reverse or a backwards mutation. So it's fairly straightforward question without going into auxotrophs or prototrophs or the way that we write them. There's little bits of the way you put sal with a minus and an H, and little bits of the way that you put it on let's say a scientific paper or a chart. So we look at mutants to wild type again being the so-called prototroph, the one we see most likely in nature. So E. coli prototroph. Here's how so when you have that little negative sign, it can't make tryptophan. Okay. Or streptomyces, or you have an R, a big R, means it's resistant to a particular antibiotic. So if you see those types of things, you'll know what they mean. I don't particularly, I'd rather spell them out as opposed to try to, I'm not so great putting those things in on, using Word. So at least it's different. My mom is a professional typist, so she typed all my papers. It's great. So this is the nugget about spontaneous mutations. I talked about this before. Occur randomly at infrequent characteristic rates. So you might recall, there's a fairly wide range. For us, it's probably 1 in maybe a billion. So that'd be like 10 to the minus ninth or more, maybe 10 to the minus eighth, maybe 1 in 100 million. But in bacteria, it's not unusual 10 to the minus fourth to be 1 in 10,000. And like I said, again, when we induce them, that we can increase this by a factor of 1,000. So that'd be 10 to the minus 1, what we call 1/10. That's a very, very frequent way. So there's a lot, and it depends on who you read is what the numbers are, but you can see there's a fairly wide range, 1 in 10,000 to 1 in a trillion for any given gene. But understand that mutations, assuming that they are not corrected by whatever mechanism the cell has, become part of the DNA. And so they can be passed on to progeny. So once it's mutated, it'll stay mutated unless there's another mutation that changes it back or eliminates it, etc. So, and this is what we talk about, a reversion or what they call a backwards mutation. And again, spontaneously at low frequency. We'll get more about that. Okay. So in a very, very large population, like in the lab, one of those little dots is a colony on a plate. It's got at least 1 million bacteria. They're not all going to be identical. We're going to have some oddballs in there. Okay. Sort of bacteria mirror life. We have a lot of oddballs out there. Okay. I'm proud to be one of them. So the environment doesn't cause mutations per se, unless it's highly toxic or, something in the environment is leading to the mutation. But the whole idea of descent with modification or evolution is cells that grow better or organisms survive more. An organism that's mutated to become resistant to an antimicrobial medication will certainly be the dominant one in an environment where the medication is present. because if it's resistant, it's more likely to survive. And that is exactly what has occurred. And you'll hear me say it about 63,000 times, what we've done to Staphylococcus. We've evolved it, not once, but twice, okay, to now be resistant to most even sophisticated or advanced forms of penicillins, even forms that were designed to combat its resistance. And that's where MRSA has come from. So understand that that's part of the reason when we look at certain agents that have the, certain biological agents that can cause problems and are susceptible to antibiotics, some of them have a greater tendency to mutate in and of themselves. So we tend to use more than one antimicrobial agent. So by definition, when we talk about tuberculosis, which we really spend most of our time with in respiratory diseases, it's mandatory to treat that with two if you're treating active tuberculosis. Mandatory, and sometimes more than two. And it's not just confined when it comes to certain kinds of viral agents, HIV being the poster child. Nowadays, we treat that with not one, two, or three. It used to be three, now it's four antivirals because of its great tendency to mutate. So you're trying to address that potential problem. So a chance that a cell become resistant to both, or an entity will become resistant to both, is a product of a mutation rate for each gene. So these are the ones we see most commonly. These are the so-called point mutations, an isolated error in the chain. So when it starts, whether it's an odd base, and you begin to see what happens. Again, the A's, the T's, the G's, and the C's. So here's DNA replication, and all of a sudden, where there should be an A, magically there's a G. Oops. Okay. At that point, you conceivably could have a mutant form and the original form as it tends to replicate. So it's one isolated error. Now it depends on where the error is and whether it causes damage or not. So the most common types that we see that you and I would get, just as we replicate our billions of gastrointestinal cells every day, is probably a point mutation. And most times they're not problematic. And if they are problematic, our immune system has ways of dealing. And all of this is sort of the backstory about keeping yourself immunologically healthy. The things that make you less likely to get sick or get diseases are to me, it's always your overall health. Whatever that constitutes, whether it's eating properly, exercising regularly, being careful with medications you take, blah, 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 all that type of stuff. And there's a lot of things that you can do. Healthy individuals just don't get sick that often. I mean, there's going to be exceptions to every rule, but those are the kinds of things that do it. So if you can do away with the really bad habits, you're probably doing pretty well. I mean, I think we've seen that, just to narrate on a little bit about it. Look, my folks were both 3-pack-a-day smokers. I was a 2-pack-a-day smoker. Okay. If I hadn't stopped, I probably would not be giving the lecture. I'm just guessing. Okay, today. But I mean, depending on when you stop, and if you get insult to injury by drinking or doing other types of substances, it's an accident looking to happen. That's the way it is. Okay, like if you were an Eagles fan, you probably were driven to drink on what was what was a holding penalty. Just saying. Sorry about it. Why do they call it? Because it's a penalty. Base substitution. So it can be silent. They have a fancy name, synonymous mutation. So it doesn't have an impact. And frequently, why that occurs is we see it in that last, when you have the transcribed codon. Now because you see the U's, we're into RNA. At that point, and the genetic code is based on the language of RNA, meaning U's instead of T's. So it hasn't really changed it. The U and G make it. If it turns out, if missense would be a substitution of a different amino acid. As I mentioned, sometimes that's inconsequential, sometimes it's moderately consequential. That's there. In the case of sickle cell hemoglobin, it's significantly consequential. And then the worst possible one is the nonsense mutation, and that's why they picked U and G, because UGA is one of the stop codons. So it will stop the process of assemblage of the protein and Proteins that aren't complete basically don't function. Hence the so-called nonsense mutation. Okay. So if it inactivates the gene, they have a fancy term called a knockout mutation. Base substitutions are common in aerobic environments. This gets back to the reactive oxygen species, the toxic derivatives of oxygen. That's really the framework of modern cancer theory and modern mutational theory. Okay. And so, as you can see, often this pairs adenine instead of cytosine. So we're looking at guanine as being one that's involved perhaps more frequently than others. When we start to see these frame shift mutations, these are either taking away a base or adding a base. Everything what we call downstream from it changes because it's in let's like I said, 3-letter words. So you could, with that little one I was telling you about before, fit cat versus fat cat. I mean, one little letter could change it, maybe not change it dramatically, but once you take away that letter, everything after that starts to make no sense. And that's really the big problem. So when we see additions or deletions, and furthermore, when you learn about various genetic diseases or chromosomal diseases, whether it's trisomy 21 or Down syndrome or Cri-du-Cat or one of the others which are involved, when we begin to see pieces of missing chromosomal material, generally that's a very bad outcome. I remember my first wife was doing, when she was approaching retirement, was taking care of a baby, like almost like full time. It was there. They had almost 24-hour care, and this poor child, had one of these genetic abnormalities that wasn't going to live very long, unfortunately, but required 24-hour care. That's not easy, by the way. And she was basically started as a peds nurse, her career. So 3 pairs change 1 codon, 1 amino acid more or less, depends on its location. I'll buy this. The ones that do this frame shift very frequently will get to. So it'll change it dramatically as, by whether you're adding or deleting, depending on what it does. And then we'll get to the transposons, and I'll show you the animation that accompanies it. Pieces of DNA that can move from one location to another in a cell's genome, and we'll take a look at that and what's involved with it and whether it blocks it or not, etc. It just depends on where they put it into. Other than radiation, so really we look at radiation being a physical agent, so whether it's ionizing or non-ionizing, we will get to, versus an induced mutation that's due to chemicals. And chemicals that cause this are referred to as, just like radiation, ultraviolet or X-rays or gamma rays, we have chemical mutagens. Most of these textbooks go through these kinds of things that are there and go over the different types that are now, and most of them are used in the laboratory when we're trying to induce mutations to analyze them. So I don't really go crazy about what chemicals are there that causes. There's just so many of them. So, and really the ones that you're going to see that are very interesting down the road for us are base analogs. And really it's the foundation for cancer chemotherapy. The vast majority of cancer chemotherapy and the vast majority of antiviral treatments that are effective are in the form of base analogs, what I like to call look-alike drugs. And you will see this again and again. So we take something that an organism is supposed to use and we tinker with it. We substitute something that's a toxin. Okay. And at that point, it doesn't work. One of the early, I'm going to tell you what it was. They dealt with uracil. Maybe I told you this before. And what they did, they know that halogens, there we are on the periodic table, are nasty. So they basically halogenated various kinds of nucleotide or nucleoside analogs. So by adding that halogen, you made a toxin that got incorporated in the chain. And so the first one that they utilized in the laboratory was 5-bromouracil. But one of the early treatments for breast cancer was 5-fluorouracil. Okay. And so, and we used to joke around because everybody said we started Mr. or Mrs. So-and-so on 5-FU. Now, you don't want to joke about cancer drugs. It's not funny, but thought I'd share that with you. In a medical environment, when you're dealing with diseases and people dying, humor is strange. You will find those of you who have relatives in the business will find it's strange. You joke about the worst possible things. So these are types. So there's different ones that, whether they alkylate, adding an alkyl group and do something like that. The base analogs resemble them, and they're going to. Here's the 5-bromouracil. Okay. Okay. So the normal base would be thymine. The 5-bromouracil. You can see this methyl group is. They replaced it with a bromine, so it changed. It looks the same, so it's incorporated. The more of this stuff you put into the environment, the more likely it's going to be incorporated, okay, and going to create problems. Or the same thing, we see adenine analogs that are there as well. Okay, just this, and so that's really where a lot of therapy is. Intercalating agents are interesting in the sense they sort of get in between. And when you get in between it doesn't allow the chain to elongate, pushing nucleotides apart, basically producing spaces. So I'm just trying to give you some context for how they're used, but not necessarily going to ask you questions. These are important. What ultraviolet light does. Ultraviolet light produces, classically in bacteria, thymine dimers. Technically speaking, it's actually pyrimidine dimers. So it can be 2 thymines or 2 cytosines, but the one we see mostly in bacteria are 2 thymines. And what happens when thymines are next to each other, ultraviolet light tends to make this occur. They cause a bond in between them. And you can see what happens to the backbone on that sugar phosphate backbone. It kind of, and again, what's DNA? Nothing more than a biological zipper. And so this is a zipper that's broken. The zipper's got a piece of material in it, something like that. And it's happened to all of us. That's kind of what happens. It distorts the molecule, distorts replication and transcription because it can't open and close effectively. There are a lot of mechanisms, and what happens is the mechanism that bacteria rely on is called an SOS mechanism, which is not terribly effective. We have mechanisms in our eukaryotic cells that deal much more effectively with thymine dimers. So that's why ultraviolet light is less effective. Ultraviolet light doesn't penetrate terribly well, but basically it can stop things from replicating and growing. That's why it's used in the food industry and sometimes in water treatment a lot. Okay. Where X-rays and gamma rays actually cause the strands to break. That's what and the difference between ultraviolet, which is considered non-ionizing radiation, and gamma rays or X-rays, which are considered ionizing radiation. It's just more damage. And then they'll get back to the transposons, and that they want to make sure we are aware of it. So spontaneous and mutagen-induced damage to DNA, cell death, cancer in animals. So 2 genes associated with breast cancer code for DNA repair enzymes. And those are the very famous BRCA1 and BRCA2, which you're going to learn about a lot, I'm sure, in your courses. And you'll probably get a fairly good introduction of that in AP2. Mutations in either result in a high probability of breast cancer. If people have both of those genes, then that's very, very significant. So they're just kind of showing you, this sort of a laundry list of things that can occur. Ultraviolet light, we have something called light repair, we have dark repair and SOS repair. We have a lot more capability and energy resources to deal with it, where bacteria do not. And then this is one I talked a little bit about before, proofreading and mismatch repair. There are a variety of enzymes that do this called DNA polymerases. They're not all equally effective. Our eukaryotic cells use this sort of magical DNA polymerase 3, which has the ability to identify the mismatch and repair the appropriate one, where bacteria is much more limiting. They just have that mismatch repair where they can basically are as likely to remove the errored strand as the appropriate strand that's there. So you say DNA polymerases, the advanced ones, can back up, remove the incorrect nucleotide, insert the correct one. Again, not necessarily perfect, but much more efficient than the classic mismatch repair that are there. And they go through all the things with regard to mismatch repair and how it's done and the enzymes that are involved, etc. This is the photoreactivation light and excision or dark repair. Okay. And again, there's not a whole lot in there and I don't really feel the need to go into the specifics. In a laboratory, you don't do this unless they've changed the way the labs are. Depends on, I've been at different schools where they've done different micro labs, where they've actually tried to use direct selection, where they've done laboratories that are involved in trying to, so they create a media that has a particular antibiotic versus one that does not, and they're able to plate this out. And I'm sure our advanced students, the bio majors probably do a lab that does something like that. And again, they use something like this. They use like a sterilized velvet material, interestingly enough. So I've seen that done. See, and they add different chemicals, penicillinases. And this is now we're getting to the end. And the last part of really of covering this chapter is coming up now. And that's really the way that we see the lateral or horizontal gene movement. But here's the carcinogen aspect that I was talking about before. Carcinogens cause cancers. Most of them are mutagens. And in other words, the way they do business is they work on DNA. And so for the longest time, they did these, and it was really very, very harsh to animals. Okay. And they began to look at this and say that mutagens increase the low frequency of those backwards or reversion mutations. So they developed this called the Ames test. And here we are, the Salmonella auxotroph, which happened to require histidine. Okay. Uses direct selection on particular plates. Okay. So the only thing that they needed to make this work to simulate this in the laboratory was the requirement, since rat liver extract, okay, a lot of these carcinogens required animal enzymes, and enzymes are very abundant in liver extract. So what they would do, they would take effectively a mammalian extract, in the case of rat liver, and grind it down. And hey, there's a lot about PETA, but they don't seem to include rats all the time. Just saying. We had to sacrifice a few rats. Oh, who cares? They're rats. I think there's an issue with that there. If they had been using cat or dog enzymes, there would be an uproar. But rats, who cares? I don't mean to be flip about that, but you would think if you're on the ethical treatment of animals, rats need. Somebody might have a pet rat that they're very fond of. Okay. And so what happens is the idea of what happens, bacteria requiring histidine for growth. You can see that histidine negative, and they plate them out. Liquids containing the suspected mutagen. And the question or not, whether or not, if the chemical is a mutagen, then we're going to see a lot, versus if it's not a mutagen, we're not going to see a lot of growth. That was the idea behind it. So if you pass the Ames test, basically you didn't make a lot of reversions. Okay. So, but if you made a lot of these reversions, okay, where the salmonella would grow rapidly, that indicated that this was an agent that was potentially a very powerful mutagen and probably should not be utilized. And so that really was sort of the way it came about it, which brings us to really the last part of that. And there's several animations that we use to look at the way that these genetic components are able to move. And that's kind of what we're going to be dealing with for the rest of today's program. So we start with, and these are in order of frequency with least frequent transformation. We use this a lot as part of genetic engineering. Transduction, which involves viruses, and this is sort of a little introduction to how viruses do business, particularly viruses that affect bacteria, and the one that certainly is it happens the most frequently, called conjugation, which involves a direct contact. So they'll show you some of these. DNA transformation involves something called naked DNA. When bacteria die, you might remember in the early parts of this chapter, well, we looked at an illustration on Friday that showed DNA exploding out of the cell. Once it explodes out of the cell, everybody, it fragments. Those fragments eventually are going to break down, but in the short term can be gobbled up. Okay. And sometimes they're in the environment and they can go to a different cell. Sometimes those fragments can be gobbled up by a virus. And those are examples of what happens in transformation and transduction. So I try to not use the word transform a whole lot. So transferred DNA replicated, okay, under certain circumstances. It can involve actual chromosomal transfers or plasmid transfers. You're going to see something called homologous recombination. It's very interesting. It's as though that DNA fragment knows where to go. It doesn't go at the end to cause a mutation. It's like it kicks out the same place on the DNA strand where it was originally located and creates a change that's there. So it's very, very interesting. Transformation is something we have been looking at before we even knew the complexity of DNA, and we didn't know why it occurred. So it involves what they call the uptake of naked DNA, DNA that's not within a cell or a virus, originates from a cell that has burst or has given out the DNA. In a laboratory, we can add enzyme, generically called DNase, that will stop it. And certain cells, when we utilize this for genetic engineering, we have to create something called competency. So that's another term that you should be familiar with. You do this by adding calcium salts and changing temperature relatively rapidly. So by rapidly freezing and warming and repeating the process, usually in the presence of calcium chloride, you're able to create a certain amount of competency. Read that as like the ability for another cell to swallow up or uptake some of this naked DNA. Most take up DNA regardless of origin. Some take DNA. So there are some bacteria, let's say E. coli will only take contributions from other E. coli. I don't know if that's true or not, but some bacteria are very species specific and others are not. Okay, so there's a lot of nuances with this, and this is more from the laboratory part of it. And I'll show you this in a second, but I'm going to go fast forward. 1920. You might recall Watson and Crick's research wasn't done until about 1948 and didn't receive a Nobel Prize until 1953. So they didn't know it was DNA when Griffith did this experiment. Down the road, there were a couple of researchers named Hardy and Chase about 1960 who were able to fluorescently label DNA and proteins to determine whether this was a protein moving or DNA moving. Obviously, it was proven that it was DNA moving. But this was really an interesting process that was done. And unfortunately, you did it with mice. So if you're a fan of mice, close your eyes. And so he was working with a bacteria that about a third to a half of you normally have in your respiratory tract, called Streptococcus pneumonia. Okay, it's the leading cause for most of us for things like conjunctivitis and middle ear infections and sort of bacterial sinus infections. So it's in there. We have it. So it's not unusual to have it. And the key is encapsulation. So he was able to study this encapsulated bacteria. And when he had that and grew it in a culture plate and injected a susceptible organism, the unfortunate mouse, the mouse got pneumonia and died. Capsules are the big player here because capsules are hard to phagocytize. And you'll see capsulated bacteria play a role in significant. Strep pneumonia is still the leading cause of bacterial pneumonia in our population. So when he had non-encapsulated cells of strep pneumonia, same bacteria, slightly different because it didn't make a capsule, the mouse kind of went, huh, I can survive this. So what happened was then he took and he heated the encapsulated cells and killed them and injected them, and the mouse was fine. Okay, so we have, look at the first thing. If we injected the living encapsulated cells, the mouse died. If he injected the non-encapsulated or the heat-treated encapsulated cells, the mouse survived. But when he mixed them together, herein was the problem. He took the heat-killed encapsulated cells and the living non-encapsulated cells, two things that hadn't caused damage in mice, but when he mixed them together, the mouse died. So he reasoned, particularly after he was able to cultivate from the dead mice encapsulated cells were isolated, that there was something in the dead non-encapsulated cells or in the heat-killed encapsulated cells or their combination that caused them to encapsulate. He was able to transform the non-encapsulated cells from the heat-killed encapsulated cells and therefore produce living encapsulated cells. He just didn't know whether it was a protein or it was DNA. Down the road, when you're able to radioisotope label it, because proteins have sulfur and DNA has phosphate. That's how Hardy and Chase did the experiment. So I will show it to you so you can kind of get an idea on how this all transferred. So we have this, and I will load these animations. I had them on a previous PowerPoint. So whether or not it works easily enough from you're just getting the zip file or not, I don't know, but we'll see. So I use the MP4s, the shockwave thing we don't see a lot of.
Involves the transfer of naked DNA into a recipient cell. In the first step, double-stranded donor DNA binds to specific receptors on the surface of a competent cell. One strand of the donor DNA is degraded by nucleases while the other strand enters the cell. The single-stranded donor DNA pairs with an homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. If there are any differences between the nucleotide sequences of the donor and recipient DNAs, the mismatch repair system comes into play. The repair system removes either the donor or the recipient strand and replaces it with the complementary sequence. Since either strand may be repaired, some cells contain the new donor DNA and others have the original DNA sequences. In the laboratory, cells are plated on selective media so that only the transformants will grow.
So what they're showing you in this is fairly straightforward. You have this naked DNA. They're suggesting that a cell died that had this capability of ampicillin resistance, for argument's sake. That way they can, and you can do this easily enough in the lab because it's easy to embed ampicillin and dissolve it when you're making up a culture plate. It's not hard at all. So here you have, now, some things that needed to occur. One, you have to have receptors for it. One, you have to have the DNA. So in comes the DNA. It's, even though it's double-stranded, only one strand enters. Not a big deal. And this is that first thing, homologous recombination. So there it is, homologously recombined. Recombination is something whenever DNA is changing places, and so we use the term a lot. So homologous recombination means it just picked up in the same spot where it kind of came from. Now you have two bacteria that effectively are identical. One has, so for argument's sake, you have one where it has a mutation and one where it has the normal bacteria. This is mismatch repair. Mismatch repair is as likely to remove the DNA that shouldn't be there as it is the DNA that should be there. Okay. It doesn't know any better. It's mismatched. So the likelihood on one or the other occurring is about 50-50. When we do this in the lab, we radioactively label it. And typically, if you have like 20 plates, you usually get somewhere between 8 and 12 plates that have been effectively recombined. It's just the way it works. So it's interesting in that regard. And so, how do we determine in the lab? We radioactively label it, we plate it out on special media, and that's kind of how transformation. It doesn't occur that often, but we utilize it a lot in genetic engineering. So, that's the first little nugget. So, as we go back and look at the PowerPoint itself, the next thing that we see occurring, so I'll back up just a little bit to show it to you. The cell has to be competent. The single strand enters, it binds, homologous recombination. And if it's not addressed, then we're going to see mutants down the road. The next one that occurs in order of frequency is called transduction. It may surprise you, but there are special viruses that only affect bacteria. They impact us indirectly. And they're called bacteriophage or phage viruses. So we nickname them phage for short. We're going to look at them in the next chapter. So this is a little introduction on kind of how viruses go about doing business. Phage infects the bacterial cell. It attaches to it. And it really what happens is it takes over. In the process of taking over, it may, when it's trying to replicate more viruses, latch on to some of the existing fragmented DNA and inadvertently in a population transfer. When we say this happens, 1 in a billion or 1 in a trillion times, the reality is that on a bacterial plate, I mean, if you got a colony, you have 1 million organisms at least. You could have a plate that has billions of organisms. So the fact that it sounds like a big number to use, where it's an infrequent thing that will occur in the realm of bacteria, it's not. That's the interesting part. And there's two different types. One's called specialized, one's called generalized. We're going to look at the generalized form of it and look at the animation that occurs. And it kind of gives you a little hint on how bacteria go about doing business. So we'll go to the next one that's there. So we probably won't get the conjugation today. We'll just use this generalized transduction.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosomes, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
So it may seem like it's a pretty interesting process. There's a lot of steps that are involved, but the reality is that this plays a significant role. A lot botulism toxin happens because of this. It's altered the bacteria and other toxins. They're carried this way. So you're going to see this. It's got a fancy name. It's called lysogenic conversion, and we're going to see it in the next chapter when we deal with how viruses work. So it happens more frequently than you think that are there. And this is really a little primer, if you will, on how viruses work. So viruses, now it's a little bit different when there's viruses that infect us versus viruses that infect bacteria. Again, they have to have a receptor, and in red, that's the viral genetic material. When it attaches to the receptor, effectively the enclosure for bacteriophage viruses is more or less a sophisticated syringe. It just injects the genetic material in. Once it's injected in, now basically what viruses do, they take over. They harvest or harness rather the host cell's machinery and create a little factory inside of the cell to make more viruses. So they fragment the existing DNA and then they go about expressing themselves. How do they express themselves? They make more viruses. Nothing else. They take the host cell's machinery, metabolically speaking. They make more what are called phage heads, viral enclosures, and they make more viral genetic material, whether it's DNA or RNA. In this case, let's assume it's DNA. And so you can see in red, it's making more viruses. Eventually, those viruses are going to accumulate to such an extent they reach something called a burst size. And so it isn't like one makes two, makes four, makes eight. It's one makes 1,000, and 1,000 makes 1,000,000, and 1,000,000 makes, you get the idea. That's how viruses in a couple of days can kill somebody. Inadvertently, because the virus is just replicating, it's not, there's no way it knows what it's incorporating. It incorporates some of the host cell's gene fragments inadvertently. When it bursts, some of the viruses are not really viruses anymore. They're really a vector, a biological conveyance to carry genetic material from one bacterial cell to another. So they really are a microbial vector, if you want to think of it that way. Then they inject the DNA combines, and again, it's subject to mismatch repair. So basically, at that point, it's transformation. DNA has been acquired, it can either incorporate or not. If it incorporates, great, then it'll go on and be genetically stable and go on from here. That's kind of the saga of transduction. The one that happens to most we'll deal with next time. That's this guy called transformation. So we'll take a look at conjugation and transfer of the F plasmid. That's the one, and I'll post these for you so you'll be able to see this one when it occurs, because this is the one that happens the most. And this is the one we tinker with for genetic engineering more than any other. Thank you. You still doing your raffle?
Yeah.
All right.